My name is Daniel Kim. As I said, I'm an assistant professor at UC Santa Cruz, also an associate faculty at the Canary Center at Stanford. So I'll be telling you about some of the work that my lab has been doing to really develop this RNA liquid biopsy technology for precision health. And so as many of you know, the human genome is comprised of over 3 billion base pairs, and much of that is actually transcribed into RNA. But what's really interesting is that if you look at the sequence space of the human genome, only about 1 to 2% of these 3 billion base pairs encodes proteins. But most of the RNA that's made is non-coding, so it doesn't make protein. And so we're really interested in understanding this non-coding RNA landscape of the human genome, especially in the context of cancer early detection. So we know that for a lot of human cancers, these RAS genes are implicated in a lot of these cancers, such as pancreatic cancer and lung cancer, colorectal cancer. So the very simple question that my lab is trying to address is, how does this mutation in RAS regulate this non-coding transcriptome, and how can we leverage this change in this vast transcriptomic space, all of these non-coding RNAs that are being transcribed, to really identify the signatures of cancer in the blood. And so what we did was we initially did this very simple experiment in a dish where we take lung cells, we introduce mutations in RAS, and then we do RNA sequencing to identify all of these non-coding RNAs. And in particular, we wanted to really look at the vesicles that are being released from these cells that are also releasing this non-coding RNA information so that we could identify these non-coding RNA signatures of mutant RAS. And so here what I'm just showing you is the size of these different vesicles. So this is a nanometer scale. So these vesicles are typically around 100 to 200 nanometers in diameter. And so you can see that in the healthy cells versus the mutant cells that have cancer, that you can see that there's a shift in the size of these vesicles. But what was really more interesting and quite striking is that if you look at the RNA that's in these vesicles, so here what I'm showing you is something called a volcano plot. Each of these dots represents a single type of RNA within these cells or vesicles. On the right I'm showing you all of the extracellular RNA that's being released by these cells. And then on the left is all of the RNA that is retained within these cells. And so in these mutant RAS cells, these cancerous lung cells, you can see that there are tons of these non-coding RNAs, what we call long non-coding RNAs or link RNAs. There are thousands of these that are preferentially being released by these cancer cells. It is giving us information about this mutational signature within these cells that we hope to harness and leverage for cancer early detection. And so we wanted to see, is this also true in patients? And as I mentioned before, pancreatic cancer patients typically have mutations in KRAS, and this is an initiating mutation that leads to the formation of cancer. And there's actually no good way to detect pancreatic cancer at a very early stage. So we wanted to see, if we looked at these non-coding RNAs, could we identify signatures that would allow us to detect this cancer very early before it's had a chance to spread, where we could help these patients hopefully survive. And so here what I'm just showing you is the different types of RNAs that are being released in healthy individuals in the blood versus pancreatic cancer patient individuals in their blood as well. And so you can see that these non-coding link RNAs are expressed and released in these pancreatic cancer patients in the blood. And what is quite striking is, here I'm again showing you a volcano plot. So on the left, these are all of the RNAs that are enriched in the individuals that are healthy. So this is healthy blood that we're sequencing. And then here on the right, what I'm showing you is all of the RNAs that are enriched in these pancreatic cancer patients. And so if you look at the RNAs that encode for protein, there's a kind of similar distribution where there's some that are enriched in healthy people versus some that are enriched in pancreatic cancer. But what was really quite striking and amazing is that in these non-coding RNAs, they are preferentially released from these pancreatic cancer patients. And we think that this will be a very strong and robust signature that we can identify in the blood to do cancer early detection. And so the vision and the hope is that we can develop this RNA liquid biopsy as a diagnostic for precision health. And in fact, all of the cells in our body are releasing these packets of information. So these extracellular vesicles that have little bits of RNA information, a lot of it is, again, these non-coding RNAs, such as link RNAs. And the nice thing about these RNAs is that a lot of them are tissue specific. So if you see these RNAs in the blood, then you can attribute these RNAs to a specific part of the body, this so-called IP address, where it gives you positional information about where this is happening in the body. And so we just received a DoD grant with Stanford, where we're going to be doing this in lung cancer patients, looking at their blood to identify these very early signatures of RNA, non-coding RNA. And we hope to do this for many different types of diseases, also for Alzheimer's. We've done this for COVID-19, and there are very interesting signatures for COVID-19 patients that will hopefully help us to understand the disease pathology as well. And so with that, we also want to do this in a handheld manner using these nanopore devices that were pioneered at UC Santa Cruz. So I just want to thank my lab. If you want to contact me and learn more about this, please email me, daniel.kim@ucsc.edu. And if you want to visit UCSC, please feel free to reach out as well. So thanks very much for your attention.